Hello, my name is Tim LeCras, and my research lab is at Cincinnati Children's Hospital. This presentation will address the role of the NRAS Q61R mutation in the pathogenesis of Kaposiform Lymphangiomatosis. The other investigators involved are listed, including Drs. Elisa Boscalo and Poonam Malik, whose labs played a key role in these studies. We are very grateful for the funding from the Lymphatic Malformation Institute for this work. Kaposiform Lymphangiomatosis, or KLA, is a rare lymphatic disorder, but patients have a poor prognosis, with only 51% survival at 5 years and 34% overall. Initially, when this disease was first described, it was thought that KLA may be a more aggressive form of generalized lymphatic anomaly. But more recently, the identification of different somatic mutations in these diseases suggests otherwise. KLA is characterized by diffuse multifocal lesions comprised of focal areas of Kaposiform or spindled lymphatic endothelial cells, accompanying malformed lymphatic channels. And you can see that in the upper right-hand panel here, which is histology of lesions from a KLA patient. Hemorrhagic effusions, particularly in the lungs and pericardium, may be involved. And in the lower right-hand panel, you can see a CT scan showing pulmonary effusions in a patient. Patients often suffer from consumptive coagulopathy and hemorrhage, which can be a major cause of morbidity and mortality. In 2018, Barclay et al. Published a study where they identified the activating mutation PQ61R in the NRAS gene in tissue from the lesions of 10 out of 11 KLA patients and 1 GLA patient with overlapping features. NRAS is a proto-oncogene in the RAS gene family, and it encodes a small GTPase that regulates cell proliferation through MAP kinase and PI3 kinase signaling pathways. NRAS mutations occur in tumor cells and increase MAP kinase and PI3 kinase signaling, resulting in unchecked proliferation. The NRAS Q61R mutation is found in greater than 20% of melanomas, as well as thyroid cancer and other tumors. Based on this finding of this NRAS Q61R mutation in the tissue of KLA patients, and the pathways known to be downstream, we propose a hypothesis shown here. The schematic below outlines this hypothesis, where hyperactivation of MAP kinase and PI3 kinase mTOR signaling pathways by NRAS Q61R drives the endothelial cell spindle morphology, increased cell proliferation, and migration of these endothelial cells into other tissues, leading to malformed ectopic lymphatic vessels. To test this hypothesis, we constructed lentiviral constructs that were generated with either the wild-type NRAS as a control, shown on the left, or the mutant NRAS gene shown on the right. These constructs were then packaged into lentiviral particles. Expression of these genes is turned on when the antibiotic doxycycline is added to the cells transfected with these constructs. Lentiviral particles containing the constructs were added to the cell culture media of human umbilical vein endothelial cells, a commonly used endothelial cell type used to model angiogenesis. Approximately 1 viral particle per endothelial cell was added. Cells that had taken up the constructs were selected for by culturing the cells in media containing puromycin, as the lentiviral constructs also contain a puromycin resistance gene. These cells were used for the in vitro and in vivo studies listed here. Here you see pictures of the NRAS mutant and wild-type endothelial cells. On the far left, not on dox, and then 24 and 48 hours after doxycycline was added to the media. You can see that the cells expressing the wild-type NRAS on the bottom retain the cobblestone morphology characteristic of endothelial cells in culture, whereas the mutant NRAS expressing cells become spindle-shaped at 24 and 48 hours on DOX. From the introduction, you remember that spindle-shaped endothelial cells are characteristic of the lesions of KLA patients. Here you see graphs of the circularity index of NRAS wild-type and NRAS mutant cells, showing a significant shift in the morphology of the cells expressing mutant NRAS on DOX in red. Here are Western blots showing expression of mutant NRAS protein, phospho-ERK levels, phospho-S6, as well as total ERK and total S6, and acting as a loading control. The increase in phospho-ERK levels and phospho-S6 in the mutant cells on DOX indicates hyperactivation of the MAP kinase and PI3 kinase signaling pathways in the NRAS mutant cells. Proliferation and migration of mutant and wild-type NRAS cells was studied using an incucyte system and migration chamber slides. NRAS mutant cells in red showed enhanced proliferation in the graph on the left, and also increased migration in the graph on the right compared to wild-type NRAS cells in green. Dr. Sandra Schenk in the Boscolo group developed this in vitro angiogenesis model, where NRAS mutant and wild-type cells are seeded onto beads and then implanted into fibrin gel in culture. You can see that after 7 days, the cells expressing wild-type NRAS in green form tubes between the beads, whereas the NRAS mutant cells in red form large disorganized structures. The graph on the right shows that NRAS Q61R mutant cells form structures with significantly greater area than the wild-type NRAS cells. Next, we generated a mouse xenograft model by injecting wild-type and mutant NRAS-expressing cells suspended in matrigel under the skin on the backs of nude mice. Nude mice are used as they do not reject the human cells. The schematic in the top left shows the experimental protocol. The cells were induced with doxycycline in culture before implantation, and then the mice were fed doxycycline in their food to continue inducing either mutant or wild-type NRAS. 8 days after injecting the cells, the xenograft matrigel plugs were removed, and those are shown in the bottom panel. You can see that the wild-type plugs on the top did not contain many vessels, whereas the mutant cells on the bottom were considerably bloody and larger and had far more vessels. The graph on the right shows that the xenograft plugs containing the NRAS mutant endothelial cells in red were heavier than the plugs in green, which contain the wild-type cells. Histology was assessed by cutting sections through the xenograft plugs and staining them with hematoxylin and eosin. Pictures on the left show the stained xenograft sections. The plugs in the bottom right-hand corner containing the mutant NRAS endothelial cells on DOX had enlarged bloody channels. The graph on the right shows that the vascular area in the mutant NRAS plugs on DOX in red was significantly greater than the plugs containing the wild-type endothelial cells on DOX in green. So in summary, our studies showed that NRAS Q61R expression in human endothelial cells induced a spindle-shaped morphology, increased MAP kinase and PI3 kinase signaling, increased cell proliferation and migration, and these mutant NRAS cells formed abnormal vessels in vitro and in vivo. So in conclusion, our NRAS Q61R models can be used to, 1, understand the pathogenesis of KLA, and 2, test new drugs and treatments for KLA. Thank you, and I'd be happy to take any questions.